All right, so this is for the lab, just looking first at building the complementary strands. So this was a strand that we had to start out with, 5' to 3', and it goes out with TAC, GGA, TTAA, GAGG, CCC, ATT, AATC. Couple things to just go off real quick. According to Chargraph's rule, purines go with pure pyrimidines. So in this case, A will go with T and C will go with G, or adenine goes with thymine and cytosine goes with guanine. This is known as complementary base pairing. As well, when you're building that complementary strand, you must also realize that you have to go in the opposite direction of the original DNA strand. So if it's 5' to 3', then the complementary strand will run 3' to 5'. So this is also known as anti-parallel. So in this case, if that was the original strand that we had, the new strand is going to be 3' to 5', running at ATGCC, TAA, TTCT, CCGGG, TAA, TTAG. So with that being said, I combined the two to act as like a parent DNA. I already unwrapped it just to give a better visual representation of what's going on. But at the origin of replication, something called DNA helicase comes in and will start to kind of like wedge itself at the origin of replication. And normally this happens like within it, but just due to graphical limitations of I can't draw very well, we're going to do it at the end here. But it starts to break the bonds. So these nucleotides, they're capable of hydrogen bonding together, and that's how they stick together. So what it does is it ends up breaking those bonds, and as it goes around, I guess through the DNA, it actually breaks all the DNA apart. And so in a normal set of DNA, that's actually happening, at the same time in both directions. So it's going to end up going, like this. And this is DNA helicase. This alone, however, isn't actually going to do everything. So you got the helicase here. What's ended up going to happen is that these are going to want to hydrogen bond back together. And so what you end up having to have is something called single strand binding proteins, or SSB proteins. And these will be represented by the green dots here. And what they do is they bind to these and stabilize them so that they don't necessarily move and try to bond back together, because then it would just do what it would erase whatever helicase was trying to accomplish doing this right here. And then along with this, so you've got the helicase here again, you've got your single strand binding proteins. So just place those, I don't know, around here. So you've actually got something all as well as called topoisomerase. And so that ended up actually being a little bit farther down, a little bit before where the helicase is. And this actually prevents supercoiling or knot formation when it's, unwinding itself from the double helix. And this is actually done by producing breaks in the DNA and then rejoining the strands. And so it'll actually start to break some of these bonds, and that will help it to not get too coiled up and allow it to, not break, because when DNA breaks, there's a whole other bag of worms that happens there. But that's, kind of just the beginnings of the opening part. And so you kind of get to this point here when you start getting into, the thick of it, where the helicase is coming in right here. And if you can imagine, it's happening on the other side as well, with the helicase, kind of coming out this way as well. But, the helicase is coming over here. It's pushing all of this DNA, and, it's still going. Got your topoisomerase right here. But what ends up happening is that at these 2 points, these are the parent strands, and new DNA has to be replicated. So what happens is you have this enzyme called DNA polymerase 3. And this is used to add nucleotides to both sides of the parent strands. So you got the 3' to 5' strand and the 5' to 3' strand. And this DNA polymerase 3 is going to actually do that. One of the consequences of DNA polymerase 3, though, is that it only adds nucleotides to the 3' end or the hydroxyl group of an existing polynucleotide strand. So what that means is that unless there is something here that allows a 3' or a 3 carbon basically on the hydroxyl group to bind with, it's actually not going to do anything. It kind of gets stuck. And so luckily though, there's another enzyme that actually helps here. So what ends up happening is that something called DNA primase comes, represented by this diamond shape thing. DNA primase, what it does is it synthesizes something called an RNA primer sequence. So in this case, it's the same sequence as before. So we got ATGCC. It'll start synthesizing this RNA primer. So in this case, the DNA primer will come here, RNA primer here, and it'll start to bond like so. So instead of thymine, RNA has uracil, so you have UACGG. And that allows this to actually start happening so that DNA polymerase 3 can come in. Once that happens, it displaces this primase, and it'll actually start to bond the rest of the, begin like adding nucleotides to the 3' end. So right here, there's going to be a 3' end that happens. So actually, I'll write that in real quick, just for clarification purposes. But there's a 3' end right here. So it's going from 5' to 3'. And this DNA polymerase is actually going to start adding, so in this case, like an A, and then a T, and so forth. And it'll keep adding and adding and adding, like so. And this actually is known as the leading strand. So this will actually just continue due to the ease of how it's already 5' to 3', and it's going towards, where the helicase is moving, where it's uncoiling. It's all functioning in that same direction. It's all growing, I guess, in that same direction is one way to put it. Later, something to note is that enzymes will actually end up degrading this RNA primer, and so it'll go away. And then something called DNA polymerase 1 will come in and actually add the rest of what was here. And I'll add it with actually correct DNA sequences. So in this case, it was UACGG. It'll come back and actually add, a thiamine, and it'll add another adenine and so forth until it actually connects together. But, again, so just to clarify, this leading strand is going to be the top strand because, as it says, once that DNA polymerase 3 starts adding nucleotides to the 3' end of the new strand, this is known as a leading strand. This leading strand will always grow towards the reflection fork, moving in the 5' to 3' direction. So, again, I'll have this come out. It's moving, as it says, towards this reflection fork that's happening with the helicase. So I'll put that back on there, just for clarification. But it's moving, again, towards this reflection fork that's happening because of the helicase. And again, this is moving in a 5' to 3' direction, this leading strand. And, and this, again, this is a continuous building. It doesn't have any stops. It'll just keep going until it reaches the end where other functions happen. But in this bottom strand, it's also known as a lagging strand. But on the bottom strand, some issues arise because of DNA polymerase. Due to the antiparallel nature of DNA, what will start to, they'll start to add nucleotides that are growing like away from this reflection fork. And because of this strand of new DNA, or this lagging strand, will be built in short set segments called Ozaki fragments. So start this off again. So you got your helicase breaking it apart. And so it's going to function very similarly to the way the leading strand started. But put a 5' here just for clarification. Helicase is going to break this apart. The RNA primer is going to come from DNA primase. It's going to put it right there. So in this case, it'll kind of start functioning right here, just for our demonstration here. But essentially what's happening is that this lagging strand has to add nucleotides discontinually to the 3' end. And so it kind of has to do it in these chunks. And so how the lagging strand works is when DNA primase synthesizes again an RNA primer, as I showed. So you have this primase with the primer. And then it places it on the parental strand, which is a strand that's actually running from 3' to 5'. Then DNA polymerase 3 comes in again, and we'll start adding the nucleotides to the 3' end of the RNA primer. So in here, I guess it'll be like a T, then an A, so on and so forth, going down. But eventually it's going to hit a point, once it's completed, it's going to hit another point where there's actually another RNA primer that's happening over here, and that's where the fragment happens. And so once that happens, the RNA primer is disregarded. So again, it disregards, and DNA polymerase 1 comes in, and we'll add in that spots where this RNA primer was. So again, that was there. It degrades, and then not that one, but the DNA, here you are. DNA polymerase 1 comes in, and I'll start adding where it was before. Big thing about the lagging strand, as it's represented right here, is that it happens in a direction away from the replication fork. So it actually builds in this direction, so away from the replication fork. And so if you can imagine, you have these fragments that are built, and once the RNA primers go away, you kind of have these nicks in the DNA that don't necessarily get fixed properly. So I'm trying to do this in a way where it makes a little bit of sense. But say, for instance, that there's just a little bit of a problem, I don't know, say right here, where there's kind of this nick in between where another primer, say, was, and where this Ozaki fragment ended. And it happens between that 5' and the 3' ends of the two, adjoining fragments. This is why the lagging strand is more difficult, is because you have these nicks that need to be, combined together. Luckily, your body also has another enzyme that's known as DNA ligase, functioning by this circle that I have here. And what it actually does is that it repairs the nick to form a continuous strand. So it'll actually end up combining, everything together here pretty nicely, and, necessarily bind it. And so then it allows for a continuous strand, and this ligase will go from one Ozaki fragment to another, to another, to another, to another, all the way down the DNA. So as the leading strand kind of goes and it has this continuous formation and it goes real nice and smooth, this lagging strand, because of the anti-parallel nature of DNA, it has to have these Ozaki fragments built, and then it has to have, you know, this DNA ligase actually bind all the nicks together as it goes. But, you know, very briefly, I guess that's how DNA is replicated.